We've seen it in futuristic sci-fi movies and television shows, doctors using some sort of genetic magic to cure disease known and unknown. But that's somewhere in the 24th century, and we're far from there. Modern science now allows everyday people the ability to know more about their own personal genetics. From K through 12 education to internet searches, all the way to direct-to-consumer genetic testing. Now, these tests do advise that their patients contact a healthcare provider in order to discuss questions or concerns about the test results. But how do primary care providers feel about discussing the clinical validity of direct-to-consumer genetic testing with their patients who may be a little more informed than before? Through a systematic review, I found that almost 1/3 of direct-to-consumer genetic testing customers share their results with their primary care provider. But primary care providers question the clinical validity of genetic tests that they did not authorize. Although primary care providers are confident in interpreting direct-to-consumer genetic testing results, few felt that they had enough training to make real clinical recommendations to their patients. And primary care providers also believe that the clinical utility of direct-to-consumer genetic testing results are only as good as the actionable items that they can translate into patient care. A few governing bodies in the field of genetics have recommendations in order to facilitate the creation of an individualized healthcare plan for the patient and provider. The American Society of Human Genetics and the International Society of Nursing Genetics call for a discussion with and education of providers regarding the risk and benefits of direct-to-consumer genetic testing that they can pass along to their patients. The American College of Medical Genetics says that scientific evidence on which each test is based should be provided in a readily understandable format in order to simplify the discussion between the patient and provider. And the Association for Molecular Pathology says that direct-to-consumer genetic testing results should be clinically meaningful. What we can take away from this is that the literature right now on this subject is varied and highly incomplete. What we need is more data on the outcomes of the collaborations and the interactions between patients and providers in creating that personalized healthcare plan. Thank you.
Today, we are going to talk about headaches, really bad headaches, also known as migraines, and they come with flashing lights and colors and blind spots. Now, I want you to imagine that headache, but in addition to the throbbing pain in your head and the flashing lights, you can't move half your body. Slowly, you become weak and realize you can't speak. You finally pass out and wake up in the hospital. And what's worse, the doctors, they're puzzled. They don't know what's wrong with you. So they draw your blood and they run a lot of unpleasant tests. And then they give you medicine just in case it's an infection in your brain. The attack I just described is a type of hemiplegic migraine called SHM1. It's caused by an abnormal gene that changes the amount of calcium inside the cells in your brain. It is very rare, and because it's so rare, not much is known about it. Living with SHM1 is like waiting for an earthquake to hit at any time. This is the way my son lives each and every day. In the brain, there's a special balance between positive and negatively charged particles. That balance needs to be maintained so the brain can send signals to the body, signals to breathe, to walk, to talk. People with SHM1 have an abnormal gene, and it makes the positive and negative range of balance inside the brain smaller and easier to change. If it gets altered, it's like an explosion inside your head that sends a bolt of electricity throughout your brain, resulting in headache, flashing lights, and paralysis, just like a stroke. Most hemiplegic migraine stops there, but SHM1 gets worse. How much worse And what disrupts the positive and negative balance? That is the focus of my research. So far in the investigation, we've learned that people with SHM1 have other symptoms when they're young that could help diagnose them early. They have weak muscles when they're born, and they have trouble holding up their heads, and they have abnormal eye movements. They struggle to roll over, to crawl, and many never learn to walk. And talking can be difficult too. Additionally, the attacks are different. They have more than just the headache and the paralysis. They often have coma and run high fevers and have swelling in their brain, which can cause seizures that doctors struggle to stop even with medication. There's a lot more to learn, like what triggers these attacks so that one day they can be prevented. But in the meantime, by knowing the symptoms, hopefully more doctors will test patients earlier so that they can manage them better. This is just the beginning in the battle against SHM1. Thank you.
Have you ever had a loved one transition into an assisted living facility? I have, and it was a really challenging time. Now, add in a global pandemic to those transitions, and that may have impacted older adults' well-being. Prior to the COVID-19 pandemic, older adults often experience negative constraints to their well-being when they move into assisted living. But those older adults also were shown to be using positive adjustment strategies to ease their transitions. This was done by finding new routines, engaging in health promoting programs, and going on community outings with family and friends. However, during the COVID-19 pandemic, many assisted livings restricted community outings, family visitation, and even those health promoting programs. Without these adjustment strategies, older adults' well-being may have been impacted. I created a novel tool called the Mueller Assessment of Transition, which measures older adults' well-being when moving into assisted living. This 12-item mat quantifies older adults' well-being. So it was hypothesized that older adults who transitioned to assisted living before the COVID-19 pandemic had higher MAT scores than those who transitioned in during the COVID-19 pandemic. In this study, 69 older adults moving into 9 assisted livings completed the MAT to score their well-being. What did we find? That older adults were experiencing negative constraints on their well-being. And this was shown because those who transitioned into the assisted living during the COVID-19 pandemic had average scores of 42 on the MAT, but those who transitioned in before the COVID-19 pandemic had higher scores of about 51. This significant difference shows that older adults' well-being was negatively impacted. So where to from here? I am calling geriatric healthcare providers like recreational therapists to work hard to implement safe, innovative, and strengths-based programs to help ease older adults' transitions when moving into assisted living. This way, a new resident can truly feel at home. Thank you.
Cotton is the primary source of natural fiber in the United States and accounts annually for more than 21 billion US dollars in products and services, creating over 125,000 jobs in cotton industry. Despite advances in plant breeding and management practices, cotton yields in the United States have reached a plateau. That is why I undertook a novel approach to improve the cotton fiber yield by altering its growth habit from perennial to annual. In this effort, I identified genotypes with high expression alleles of 5 floral induction and meristem identity genes, flowering locus T, suppressor of constans 1, fruitful leafy, and apetala 1 from an upland cotton mini core collection. and cross them in various combinations to develop cotton lines with annual growth habit, optimal flowering time, and enhanced productivity. To facilitate the characterization of genotypes with the desired combinations of stacked alleles, I identified markers associated with the gene expression traits via genome-wide association analysis using a 63,000 single nucleotide polymorphic array. Over 14,500 SNPs showed polymorphism and were used for association analysis. A total of 396 markers showed association with expression traits. Out of these 396 markers, 159 mapped to genes, 50 to untranslated regions, and 187 to random genomic regions. Biased genomic distribution of associated markers was observed where more trait-associated markers mapped to D subgenome. Many quantitative trait loci coincided at specific genomic regions. This observation has implications as these traits could be bred together to develop high fiber yielding varieties. As cotton is a social fabric, therefore these findings can also benefit the community both economically as well as socially. Thank you.
Good morning, everyone. I'm Prasanna Shekar from the Department of Plant and Environmental Sciences at Clemson University. The topic for my 3-minute thesis presentation is to investigate the use of nucleic acid as pesticide to manage major pest and pathogen. Here, I'm going to speak about a pest and a pathogen. The pest is Melanophis sacchari, also called as sugarcane aphids, which affects about 90% of sorghum acreage and causes 50 to 100% of yield loss for the sorghum farmers. Similarly, the armillaria affects about 3.86 million in annual loss to the peach producers. The common current solutions which we have in case of sugarcane aphid is scouting 2 to 3 times a week, use of conventional, which is a synthetic pesticide, and resistant hybrids and integrated pest management system. In case of armillaria, we don't even have that. Either you have to do tree removal or a above ground root collar excavation, both of which are a temporary solution. The major problem with these solutions are you need to repeatedly apply the insecticide or the fungicide, which causes a major environmental problem, and it also affects the beneficial insects, wildlife, and human beings. The major other issue is the development of the resistant strain. So we need to find another insecticide or a fungicide within a short span of time. And it also causes soil contamination. We are trying to remove this problem at the root. So what are we trying to do is we are using RNA interference based method wherein you can use unmodified double-stranded RNA or coat the double-stranded RNA with a chitosome or liposome complex or use of recombinant bacteria expressing the double-stranded RNA. The main idea is to protect the nucleic acid so that it does not get damaged. In case of armillaria, you can even use RNase-based method. Now, the major impact of these two is it's not restricted to either sugarcane aphid or armillaria. You can use for any insect or pest or fungi for that matter. So this gives a broader impact of this method. We are going to try different delivery methods in the laboratory, greenhouse, and the field. The major significance of our method is it is reprogrammable, less quantity of pesticide is used, it is eco-friendly, less residual activity on the soil, and it is very target specific. These methods will create a greater impact both economically to the farmers and also it is environmental friendly, and we don't have to repeatedly keep on applying the pesticide or the fungicide. So these are the advantages of our methods. Thank you so much.
There has been increasing demand on organic produce in concern of healthy food resources and environmental pollution caused by traditional agriculture. In 2015, nearly 40% of the inorganic nitrogen fertilizer applied in US leached into the soil, which heavily polluted the surrounding ecosystem and even our drinking water source. However, high requirement of soil management has limited the productivity and market competence of organic produce. Imagine you are shopping tomatoes at a grocery store. Will you choose normal tomatoes or organic tomatoes that are 30% more expensive? For a long time, we've been working so hard on soil management without noticing one of the most important factors, soil microbes. But why do we even care about those tiny guys in soil? Well, soil microbes are actually the second largest biomass source in a terrestrial ecosystem, with up to 10 billion individuals living in 1 gram of soil. And so far, only less than 3% of the soil microbial species has been identified. Soil microbes have significant impact on agriculture, taking part in almost every nutrient cycling process in soil and protect crops from disease. With the limited knowledge of soil microbes, we have enough reason to guess the potential of soil microbes on organic agriculture. So far, soil chemical and physical properties has been well studied, but the effect on soil microbes remains vague. The main goal of our research is to understand the interactive effect of tillage, cover crops, and manure on soil microbial growth and nutrient cycling. Our major experiment units are reduced tillage and conventional tillage. Under tillage, we have different combination of manure and cover crops. Nitrogen mineralization rate, the rate at which soil microbes transform organic nitrogen into plant-available inorganic nitrogen, will be measured as an indicator of microbial activities, and the soil microbial biodiversity will be measured by 16S RNA sequencing. During the incubation experiment of the first growing season, significantly higher ammonium was found in soil grew with winter rye and hairy vetch compared with soil without cover crops. Ammonium is the product of nitrogen mineralization. So here, higher ammonium indicates potential higher microbial activities. This is a good start. We will continue monitor soil nutrient cycling and move on to DNA extraction. I hope this project could develop a management that maximizes the microbial growth and reduce the cost and increase the productivity of organic agriculture.
Hello, my name is Josh Osuofa. Today I'll be presenting on membranes as an innovative tool for the rapid production of monoclonal antibodies. Do you know someone who's ever had breast cancer or that is suffering from autoimmune diseases like rheumatoid arthritis? Then chances are they're getting treatments from monoclonal antibody drugs. For example, someone with breast cancer typically gets treated with Herceptin, and someone with Humira, with rheumatoid arthritis will get treated with Humira. These two drugs are examples of monoclonal antibodies, and they represent a rapidly growing class of drugs in the biotechnology space today. They're expected to have a over $200 billion cap in the year 2023 and going forward. Manufacturers are facing the problem of meeting this growing demand for monoclonal antibodies. So in our research, we tried to find a way to increase the production capacity and also increase the speed of production for a lot of these pharmaceutical companies. What we found in our research is that there's a specific step called the capture step that we can improve. In this capture step, the monoclonal antibody that is being produced is selectively bound to a resin column inside the pores of resin beads. Once they're selectively bound, all the other impurities are then washed off, and then they can be selectively eluted and then sent, eluted meaning they can be selectively recovered from those beads and then sent off for further processing. What we found is that in this step, the problem is as you increase your flow rate, the amount of drugs you can bind continues to go down because you're using a resin bead. What we found is that with a membrane, with increasing flow rate, we can actually maintain the same binding capacity, which means we can bind the same amount of that antibody drug. So as scientists, our job is to convince the industry to adopt the use of membrane columns. We've done that by exploring and evaluating the commercially available membranes in the space today. We found that Cytiva and Purexa can actually bind 70 to 80 milligrams of our drugs in as little as 5 seconds. That is 72 times faster than what a resin column can do. They're also reusable. You can use them up to 10 times without any loss in capacity, and they have very low pressures. I hope this shows that membranes are a very good alternative for the rapid production of monoclonal antibodies. Thank you.
Imagine a historic neighborhood, maybe one you've driven through on a road trip. Near your hometown you would go to for seasonal events such as summer fairs, or one you've seen pictures of but have not yet had the chance to visit. And now imagine if every piece of what made that town look historic disappeared. The wood shutters were gone, brick roads paved over with asphalt, and large skyscrapers popped up the next block over. The integrity behind a historic district is what evokes that sense of history it intends to provide. And now all of that is gone. When thinking about historic preservation, one first thought that comes to mind is the National Register of Historic Places. Established by the National Historic Preservation Act of 1966, the National Register creates financial and community incentives for local preservation. However, there are very few federal regulatory teeth in its legislation that actively protects these properties. They could be listed on the National Register one day and then demolished the next. It is the local stakeholders and actions on the community and state levels that provide the preservation these communities need. Some communities, such as Charleston, South Carolina, have strict local ordinances aiding in their preservation. Proposed alterations must go through a Board of Architectural Review, where the public, including local preservation foundations, have the opportunity to comment. Other communities, such as Edgefield, South Carolina, have a strategic plan for their community, but no local interventions that are codified to create this reality. This thesis will look at National Register historic districts, regions possessing a significant concentration, linkage, or continuity of sites that relate to each other historically or architecturally. Districts derive their importance from being unified entities. While many historic districts have strict local ordinances that preserve the tangible integrity, many may not. For this research, I'm looking at preservation ordinances, planning codes, and comprehensive plans for 7 communities throughout South Carolina to understand their local interventions. I am visiting these communities' National Register historic districts that were nominated between 1970 and 1983, and I will measure the loss of integrity seen over time. This study is looking at local preservation policy in a manner at a level that has not been undertaken before, and will be contributing to the understanding of the efficacy of preservation policy in local communities across America. The goal of this research is to determine if there is a projectable lifespan for National Register historic districts that lack local interventions, what that lifespan looks like, and how that lifespan can be extended. It is important for communities to understand how their preservation policies are working and how they can be improved to retain stronger integrity for years to come. Because the wood shutters and the brick roads that take you back to the 1870s will ideally last longer than the 2030s.